Hey everybody, welcome to our monthly journal club that we're taking place a little earlier today, earlier this month than usual, December, December 19th, because of the upcoming holidays. So today, the journal that we're going to, let me do some introductions. Myself, Oliver Medvedik, broadcasting live from New York City, the Cooper Union for the Advancement of Science and Art. And I'm here in my laboratory. And along with us is Steve Hill.
Yes, and I'm here live, and I'm in my secret underground facility.
I'll post the GPS coordinates to this facility in a few moments. It's somewhere in the EU. Well, actually, no, it's not in the EU. I take that back. It's not in the EU.
Yeah, we are in the EU. We are in the EU for a little while longer.
Oh, okay. All right. Well, when you pull out, let me know so we can celebrate your independence. Anyway, so the article that we're going to be covering today, let me put the article up. It's an interesting one, a very interesting one. Not that all the other articles we've covered have not been interesting, but this one's a little bit different. It talks about, puts together a variety of disparate subjects: aging, Alzheimer's, light frequencies, neuroscience. So there's a lot going on, and I'm not really an expert on many of these things, but we'll try our best. So the article is, here it is. Gamma frequency entrainment attenuates amyloid load and modifies microglia. All right, so this is a Nature paper that came out fairly recently. There's a lot of authors here. I can point out, so it's Iacarino et al. And if you notice, the last two authors are Edward S. Boyden and Liwei Tsai. So Liwei Tsai is over at MIT right now, I believe. She actually was at Harvard when I was an undergraduate. She had a lab there at Harvard Medical School. And Edward S. Boyden, some of you probably have heard of him. He is basically one of the pioneers of optogenetics, which is a technology that's used to stimulate neurons using light. And the way this technology works is essentially using specially engineered ion channels that are light regulated. So these are channels, sodium channels that will open and close in response to pulses of light. So you can use light pulses to essentially switch neurons on and off. So that gives you a hint as to some of the technology that's being used in this paper. Okay, so what's this all about? So just to unpack the title a little bit, gamma frequencies are these oscillations between 20 and 50 hertz. In the paper, they settle on 40 hertz, which are basically oscillations of neurons firing. So neurons in your brain, neurons in mammalian brains, and probably in the brains of many other organisms, perhaps invertebrates as well. They don't randomly fire. They fire in certain coordinated frequencies, and these oscillations are given names. And this particular frequency, I've just done a little bit of looking, it's associated with perhaps consciousness. There's probably a lot of other things it's associated with, but essentially the neurons are entrained, and they'll fire in synchrony at 40 hertz. And the reason they're looking at these 40 hertz gamma oscillations, just to give a little background, is that it's been noticed for some time that the neuronal firing, the oscillations in brains of people, of animal models that are pathological, in some cases, neurodegenerative diseases, their oscillations are off, or attenuated, or somehow not regulated. So this paper takes it a lot further, and they want to probe as to, what's happening? Is it perturbed? Is there more to this perturbation? Is there something that this perturbation of 40 hertz oscillation is actually doing to the brain that's bad? And the flip side, is 40 hertz oscillations something good, right? So as the title suggests, this gamma frequency at 40 hertz entrainment, meaning all the neurons are firing in a coordinated fashion, attenuates amyloid loads. So amyloids are these sticky proteins that accumulate in the brains of individuals and organisms that have neurodegenerative diseases such as Alzheimer's, and particularly beta amyloid plaques. And it seems that this gamma frequency entrainment will reduce this amyloid load, this amount of these proteins, and also simultaneously it modifies microglia. So microglia are, there's a percentage of your cells in your brain that are not neurons. Microglia are sort of like the macrophages of the brain. They do a lot of housekeeping functions, and it's about, I believe, about 15% of the cells in your brain are microglia. And they connect this microglial function with the reduction in amyloid load, and this is also then connected with gamma frequencies. So gamma frequencies somehow do good things. In other words, they promote microglia to help clear out amyloid plaques. So let's stop sharing here and show my face now. So that was just the title alone, folks. So just to give a summary of what we're going to be seeing here. Any questions? No? I think we should just go further, take it to the next figure, I guess.
Well, it's interesting you mentioned its effect on microglia. We've seen this happen before in other approaches. Alexandra Stoltzin has been doing some work on macrophages, or particular microglia in the brain, and they found out that by modulating their behavior, it does actually help attenuate plaques. Now, if you recall, I think a couple, maybe 2 or 3 journal clubs ago when we were talking about hallmarks, I mentioned polarization of macrophages. Do you remember that? Where I talked about them being M1 and M2 type? Like the M1 is like the aggressive inflammatory type of macrophage, almost like a profile, and the M2 polarization is the sort of calming, anti-inflammatory and pro sort of healing. And a lot of different studies using different approaches have found that by changing the ratios of macrophages from M1s to M2s, they've been able to reduce things like plaques in heart disease, Alzheimer's and other things. So it kind of already ties into that, doesn't it?
Yeah, it does. And one thing I also mentioned a couple journal clubs ago was there was another paper which we haven't done, but they looked at a certain cytokine, IL-33, interleukin-33, which the levels go down as we age, and injections of IL-33 have shown to stimulate macrophages in the brain to help clear out plaques. And unlike this paper, which has this paper, if there's only one critique I would do, and it's not really a critique because that's not what they were looking at. They've done a ton of work just to lay the groundwork, and I'm sure the follow-up paper is going to look at this. Is the actual, are there improvements in memory in these mice? Are there changes in the physiologic parameters of the mice? And the paper that looked at IL-33, which boosted the effectiveness of macrophages, they did look at physiological responses, and it improved the symptoms by 50%. So it would be very interesting to see, I'm sure the next paper is going to look at precisely this. What is, how are these mice performing? And all of these things that, boost the effectiveness of microglia and clearance of plaques. There's a lot of other things, that do this. And, a really, good question is, are they additive, right? Can we combine them all, or is it already saturated? Probably not, because, we'll see here in this paper that the improvement is only about, in plaque reduction, 20 to 50%, which is pretty high, but all the other ones that I've looked at is also within that range. So, and the really cool thing about this paper is, we'll see later that, first they do this experiment invasively, but there is a way to non-invasively entrain your neurons. So this is this to me is, no pun intended, the very stimulating aspect of this paper is that, you know, it's a very safe procedure potentially, and it's non-invasive. In fact, it's so non-invasive that, you know, and so simple that there's a lot of companies out there, you know, selling products that are basically lights that flicker at 40 hertz, and I'm not, you know, I'm not a shareholder in any of these companies, but just to give you an example, just, you know, just to show people what we're talking about here, there's a website, gammalightTherapy.com, and for $39.99 or $40, which I guess that's you know, supposed to be tongue-in-cheek, right? 40 hertz, $40. You get this really pretty set series of LED lights that will flicker. I don't know if you can see that flickering, but it's, you know, it's hard to see flickering because of you know, the transmissions and whatnot. But it's not as bad as I thought. This flickering is very rapid, and it's actually, I thought, you know, it'd be annoying to stare at, but it's actually kind of mesmerizing, you know. So, you know, for $40, I got this giant string of LEDs that are flickering at 40 hertz, which actually is long enough to kind of wrap around a Christmas tree. So if you want to, you know, have a Christmas tree with lights that actually might be helping you out, there you go. Anyway.
And here's to be thinking you just popped down to Target and bought a box of fairy lights.
Yeah, you got to get the frequency right. Anyway, so very fascinating stuff. So let's go back to the paper. All right. So let's scroll down here. There's a lot of terminology, a lot of methodology. They look at, I believe, hippocampal region of the hippocampal subregion CA1. I'm not a neuroscientist, so if you showed me a brain and told me where's CA1, I wouldn't be able to tell you. I'd have to look it up. But I'm going to figure 1, and figure 1 is basically, so they're using, just to kind of lay the groundwork, there's a lot of jargon here, but they're using mice. They use 2 types of mice, but one type of mouse that they look at are these 5X FAD mice, and these are these 5X familial Alzheimer's disease mice, which basically these poor mice have 5 different mutations in 5 different genes that basically really accelerate the accumulation of beta amyloid plaques. So these mice are you know, have like hyper Alzheimer's, and they have these engineered mice where you see PV-Cre times 5X FAD. Well, this is parvalbumin Cre lock system. So this parvalbumin is a protein that's found specifically in interneurons, and these mice have been engineered. This is just to kind of zoom in here. This mouse here, the little brain. I guess this is CR1. This is a channel rhodopsin CHR2. This is this modified protein that's expressed that's light sensitive. So it's basically a channel protein. And this is where the optogenetics comes in, where you can actually stick a fiber optic conduit into the mouse brain and flicker and entrain these neurons by pulsing light, you know, at a particular frequency that's, you know, I think bluish, that this channel rhodopsin protein responds to, and basically get it to turn on and off at whatever frequency you want, right? So this is the very first experiments that they did to see what would happen, and what would happen specifically with beta amyloid plaques. So let's zoom out here. Oops. Okay, so I don't really know how to read these, you know, very well. These are plots of frequency, and they have, you know, all of these different spikes happening. So SWR is, God, I have to look this up. I keep forgetting what it is. It's sharp wave ripples. So they have all these different oscillations brains can undergo. Gamma is 40 hertz. Theta oscillations are 4 to 12 hertz. Then that happens in quiescent behavior. When the animal's active, you have gamma frequencies, and then, or I guess in quiescent behavior, you have sharp wave ripples. So what they're looking at here is, so for think there's a correlation with these gamma waves, 40 hertz, with sharp wave ripples, which are at a higher frequency. I'm not sure of the significance of that, but all of this is basically laying the groundwork to show that they can measure these frequencies in their mouse model. And here is in E, the model where they're actually pulsing, and this is this pyrospectral density, which basically means, I guess, a combination of amplitudes to show that when they're doing this, 40 hertz pulsing, you get a peak of pulsing happening here with frequency. So that's showing that their system is working. They can basically get these neurons to pulse at the right frequency. The more interesting stuff is in figures F, G, H, and I, where figures F and G, zoom in on this. Oops, sorry about that. Figures F and G are in this mouse model where you have these labeled neurons, or the neurons that can be switched on and off with light, that are interneurons, and I think these are these neurons use neurotransmitter GABA, and then they have, as a control, they have another group of neurons that are light switchable. This is figures H and I, which are alpha cam K2, Cre, which are excitatory neurons. So what they're going to show here is that seems to be specific for at least this beta amyloid clearance. So they're looking at here beta 140 and beta 142, which are just two variants of amyloid, beta amyloids. So there's one thing that kind of threw me off here a little bit, is that they have, so what you're looking at here is the percentage of plaques as normalized to an EYFP control. And EYFP is just a protein that's being expressed, just yellow fluorescence protein that's being expressed in the neurons that's not, not a channel rhodopsin. And this is all supposed to be normalized to EYFP. So, so this percentage you see here is normalized to EYFP. So this would be like the negative control. I don't know why we have a bar here for EYFP if it's all normalized. EYFP normalized to EYFP. I'm not sure. I would think that's 0, but I didn't get a chance to really thoroughly dissect the methodology here for making that ratio. But what they're showing here is that if you have 40 hertz stimulation, you have, and this little line here shows that there's a significant reduction. It's a little messy, but according to the statistics, a significant reduction in these beta amyloids, and that between EYFP and, I'm assuming that EYFP here is not stimulated at all, or maybe the light's on completely, versus 8 hertz, there's no significant difference. What is interesting is that they don't really talk about this, but having random light, just flickering randomly, makes it worse. So, they show that a number of times. So this is looking at this other variant of amyloid beta, and you see the same thing. But, again, they're stressing the 40 hertz reduction. You don't see that effect with, with these other neurons. Although you do see, again, seems to go up with random flickering. So I don't know, maybe that doesn't bode well for people who sit under randomly flickering fluorescent lights. That's something maybe to watch out for, no pun intended. So I don't know. I don't know what to take from this random that seems to kind of shoot up. Maybe if you want to drive somebody crazy, you just have random lights flickering, and that will literally do it. But they initially see this reduction in beta amyloids. So just looking at the mice, these are all samples taken from mice, and they have numbers here. It's about 8 to 6 mice for each bar. Okay, stop sharing this for a moment. Any questions? Comments?
There is some comments. We have James Knowles, who said, I read a paper about cyclodextrin infused into the blood, activated, well, he says microwaves, he means macrophages, that cleared up to 40% of arterial plaque buildup. I actually wrote an article about that. It was quite interesting how this type of sugar achieved it. He was saying, might this also be a good carrier for the medication too?
You know, I don't know. Cyclodextrin is interesting. Isn't that what's in Febreze?
Yeah.
So it's these cyclic compounds that basically, what my understanding is that they capture organic compounds, so they kind of lock them in, non-specifically. So when you spray Febreze, that's what it does. It captures these compounds. I mean, the non-specific stuff kind of worries me, because it's like, you know, I mean, it's glomming onto a lot of stuff, right? So it's not just specific to like one thing. So, you know, supposedly it's completely safe and harmless, but I mean, if you're spraying this stuff all over the place, it's latching on to basically anything organic floating around, which, you know, maybe in the air is fine, but in your bloodstream, I'm not sure. Yeah, I don't really know about the specifics of cyclodextrin.
You'll have to check out the article we wrote about it a couple of months ago then, Oliver.
Yeah, well, you know, I don't remember, but if I stare at this maybe for another hour, it'll come to me.
We've got a comment about those lights, actually. Ronnie is saying that they were wondering what the decorative lights were for. I'm pretty sure it doesn't work through the eyes.
I'm pretty sure it does, actually.
Really? I thought they had to insert the fiber optic into the brain. Huh?
Well, that's how they started. Figure 4 is where the really cool stuff happens.
Because I'm picturing that Hellraiser guy now, Pinhead, you know? Except with instead of all the needles, you'd have like little fiber optics in your head. I don't fancy that.
Well, you can't see it here, but I have a trepanation port back here, and I actually feed this all into my head, and I did inject myself with a virus that is carrying channel rhodopsin, and I'm just kidding. Anyway, okay, let's get back to the paper.
It's Christmas. It's Christmas, folks. It's the time of year that Oliver decides to be a little bit jovial.
Too much eggnog. Anyway, all right. So now we have figure 2 and 3. This is a nature paper. So what do you expect? There's a lot of stuff jammed in here, and we're not going to get into the extended data, which is about 10 times longer than the paper itself. So if you do want to read this paper, there's a lot more that we're not going to cover. But anyway, figure 2 is, the title for that is driving 40 hertz oscillations optogenetically in hippocampus reduces amyloid beta in 5X FAD mice. So they look at several variants of APP protein, which is, what does it stand for again? Amyloid precursor protein, I believe, and several truncated variants, NTF and CTFs, which basically are precursors to amyloid beta formation. And this Western blot here is basically, I think it's a Western, represented Western blot. This is from 2 mice. So EYFP control, random flicker 40 hertz, EYFP random and 40 hertz. Beta-actin as a control, APP normal protein, and APP NTF and APP CTFs are the truncated variants that will lead to bad things. And we see with 40, so EYFP control. So here's here's where they actually do the normalization, normalized to EYFP. So EYFP, I'm just trying to, this is so full-length APP, there's no difference. You do see, you know, a slight but significant decrease in with 40 hertz, decrease in both of these, you know, so they're basically boxing in these bands and then comparing them to EYFP as a normalization. I don't know, it kind of looks a little, though, that when you do random, that this goes up quite significantly. If you look at random here, it says there's no difference. But if you look at this blot, I mean, I don't know, maybe my eyes are off, but this looks a lot darker than this, doesn't it?
Well, to be fair, Oliver, whenever I do look at those blots, it always makes me think, you know, it always makes me look for me glasses, and then I realize it's meant to look like that.
Yeah. Okay. Anyway, so we'll take the data at face value and say that it's a decrease. What they do here is this nice immunofluorescence. They're actually looking at so they're looking at neurons, and this is immunohistochemistry against amyloid beta and anti-EEA1. And this EEA1 is, I believe, a protein that is a marker of endosomes, and this is where amyloid betas basically get kind of processed. So EYFP, you see all these amyloid betas. Hook staining is basically nuclei, and this is EEA1. And 40 hertz entrainment, there appears to be less amyloid betas, less, let's see, make sure I have EEA1 actually explained correctly here. Where's the, sorry, scrolling through here. Endosomal labeling of CA1 neurons, both EEA1. Is there anything special by EEA1 other than being an endosomal marker? Enlarged early endosomal antigen. Yeah, okay, so there's more endosomes basically, you know, where less, or should I say less endosomes when you have, well, significantly less endosomes when you have 40 hertz stimulation as compared to EYFP control. You do see a heck of a lot more with random, though. Again, I don't think this is supposed to be the control. EYFP is supposed to be the control. So if that's the case, again, does that mean that randomly flickering light is dangerous? 40 hertz oscillations. I mean, random flickering is somehow, you know, increasing amyloid beta. I mean, that's what it suggests to me. Again, the point of this paper is to stress the 40 hertz decrease, but you know, I keep seeing this increase with random, and maybe I'm reading the controls wrong, but you know, that's what it looks like. Okay, figure 3. So then they go into driving 40 hertz oscillations optogenetically in a hippocampus, and they look at morphological transformations of microglia. So when microglia start to like eat and engulf things, basically get fat, and that's what they're looking for is fat microglia. The one thing that I was a little confused about here is in figure 3A, what they're looking at here is they're doing an RNA transcriptome analysis, so that it's RNA-seq. So they're looking at a whole RNA transcriptome, and here we see red things that are upregulated, things that are downregulated. EYFP control, when they switch on 40 hertz, you get a different pattern of genes that are upregulated and downregulated. And here they make a pie chart saying that 35% of these correlate with microglia, 19% with neurons, endothelial 17%, astrocytes 14%, and some other neurons, I'm not going to go through the acronyms, 951%. And this is their basis for them looking at microglia. I'm not sure how they managed to, initially I thought, well, they isolated all these neurons and then they did RNA-seq on all these different neurons, but then it looked like they took the entire brain region and they just took all the RNA out and then used some sort of comparative software to kind of I don't know, like some kind of Fourier transform or something to then partition this into different categories. And I'm not sure how that works. So if somebody, knows out there, I'd be happy to get some input. So I don't know how you can deconvolute which of these goes to which of these on the right. But suffice it to say, you could read the materials and methods, and they said they got a whole bunch of data sets that are publicly available that correspond to the expression patterns for all these different neurons, and some sort of mathematical statistical wizardry was then done to basically, I guess, bin these into different categories. So microglia wins out. And what they do here now is they're looking at normalized to EYFP. They're looking at specific categories, and this is interesting, specific categories of RNAs. And everything kind of from CSF1 to the right, all of these bars that are going up, these are markers of microglia that are kind of actively eating stuff, right? So you see all of this going up. Make sure I get this right. So here we say, upregulated genes included microglial engulfment associated genes, CD68, B2M, et cetera, et cetera. Interestingly, on the left are stuff associated with inflammation. And they say there's no significant changes, but there kind of are a little bit. I mean, not as significant as what we're seeing here, but if you really zoom in, IGF-1 is associated with a suppression of inflammation, and that actually goes up, goes up twofold, right? So it's significantly higher, not as radical as this, but I wouldn't call that insignificant. This IL, these are indicative of inflammatory responses, and these are insignificant, but this one might be significant, and this one goes down. So I don't know, perhaps they're being overly conservative, but I would say that, there might be some suggestion that you are suppressing inflammation. Maybe, perhaps, I'm being maybe a little less conservative with their data. But anyway, suffice this to say, they basically, show that microglia are actively eating stuff, this stuff being amyloid beta, and here are some immunohistochemistry images, very beautiful images of microglia stained with protein that's specific for microglia, stained for amyloid beta, and then merging. And you can see here, these are zooms of individual cells. These 40 hertz, you could see here that if this is the same scale, which assuming it is, they're a lot fatter. And basically what you end up showing here in these bars, and we'll look at that in a moment, is that with 40 hertz entrainment, the microglia, the cell bodies get bigger, and their processes, their dendrites get shorter, so they kind of like pull in. And that's indicative of microglia actually engulfing stuff. So here we see microglial count. It actually goes up per whatever section they're looking at. In this case, we're not really seeing any changes with random flickering, but we see an increase in microglia go up. We see cell body diameter go up. We see average processes length go down, and percentage of microglia, also amyloid beta positive, go up. So here we have a correlation. So not only, these two bits of data are basically suggesting that this 40 hertz light entrainment is perhaps operating at two different levels. One is they actually slowing down the accumulation of the production of amyloid betas, but also when amyloid beta is present, it's also stimulating microglia to devour it, right? So that's the take-home message from these two. Stop sharing here.
Well, that's quite interesting then, because that's at least a couple of hallmarks, because you've got loss of proteostasis and misfolded protein, so somehow it seems to be influencing that. So it's helping to stabilize proteostasis.
Mm-hmm.
And obviously it's upregulating, autophagy and such like as well. So that's interesting.
Mm-hmm.
But what sort of, we had a question in the chat there. So, James Knowles was asking, So what was the percentage of plaque reduction?
I should tell you the numbers as well. Let's see. We found that 1 hour, so this is going to the previous figure, which is here, FGHI in figure 1, that the interneuron stimulation reduced amyloid beta 140 by 53.22%, amyloid beta 1-42 by 44.62%, so roughly 50%, 40 to 50% reduction in amyloid beta, and gamma induction. Let's see what we have here. So with the endosomal labeling, the decrease in EEA1 endosomes was decreased by about 40%. And let's see, down here, figure 2 and 3. So microglial cell body diameter increased by 135%. The links contracted of the primary processes by about, well, okay, so they're not, I said call them dendrites, but primary processes, not dendrites. 54%. Percentage of microglia co-localized with amyloid beta in the cell body increased to 85% from 31%. So yeah, about a 40 to 50% increase in localization. So yeah, so far we're seeing, stuff on the order of 50%, 50% cleared, 50% localized to microglia. Okay, so let's go to figure 4. Figure 4 is very interesting because now we move away from actually, having to do optogenetics and having to drill a hole in the mouse's head and snake a fiber optic cable in there. And here what they're actually doing is looking at not the hippocampus, but looking at the visual cortex. And the visual cortex is accessible to the outside world through your eyeball, right? So we receive photons, hit the retina, get picked up by rods and cones, and we have a change in membrane polarization, actually a hyperpolarization.
Okay, well, I'm going live again. We've got the test card up again at the moment, Oliver.
Okay.
So I'm just going to let people know. Okay, so yeah. Are you ready? Oh no, Oliver's not ready yet. You need to disappear.
Are we on?
Well, I think we've had a slight technical hitch there. Best laid plans of mice and men, or rather Facebook being annoying that if you do drop the stream, you have to start a whole new one. How annoying is that? Anyway, we're back, Oliver.
Okay, welcome back, everybody. So we left off right before the last figure in this paper. Well, not the last figure that's in the published paper, but not in the extended paper. There's about 100 more figures. And this is the figure where we basically go away from a very artificial system of optogenetics to actually stimulating the mice using an external light source that's directed at their eyes. So they're now going to be looking at what happens when you entrain at 40 hertz the visual cortex using light pulses or flickering lights that the animal can see. And short answer to what happens is that you do get a reduction in amyloid plaques. And that is very, very exciting because it's a very simple procedure and it's a very non-invasive, and there doesn't appear to be any risk to an animal. So let's take a look at that figure. Okay, so there's a lot of similar stuff that they do here just to show that it's working. So up in figure A, they're looking at this power spectral density frequency. So this is now light flicker, not optogenetic, but just flickering lights. And again, just to give you an example, probably this is not going to come through very well, but I mentioned earlier I have this collection of lights here. These LEDs are flickering at 40 hertz. And surprisingly, it's not too unpleasant to look at. I thought I'd get a bit nauseous staring at flickering lights, but it's at a very high frequency, so you can just, you know, if it flickered a little more, it would be barely perceptible. But so they had these mice exposed to these lights flickering for about an hour, I think, hour intervals. Not these particular lights, but these I purchased. Okay. So you can see that, you know, they get, so here they're recording neuron action potentials electrophysiologically, I presume, and you can see a spike at 40 hertz. This is, I believe, in the visual cortex. So that's just showing you that whatever lights they're shining on the outside, the entrainment is happening on the inside. So this is just showing time from light flickering in milliseconds. So I think they do, what did they do here? 1 millisecond pulse at 40 hertz, something like that, somewhere in the text. Anyway, so in figure C, what they look at is the actual accumulation or not accumulation of amyloid beta plaques. Oops. Let's try to zoom in on this. I can't seem to find it. There it is. Zoom, 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 zoom. These are really tiny figures. Scroll, scroll. Zoom, zoom. Okay. So what they're looking at here is control animals. So they basically, you know, I believe, remove parts of the visual cortex and then go see what happens to amyloid beta. Two variants, amyloid beta 140, amyloid beta 142, and this is a percentage normalized to negative control, which is, in this case, so you can see all the treatments that have been done down here. It's light, so this is, I believe, just light on or off. Now light at 20 hertz, at 40 hertz, at 80 hertz. PTX is picrotoxin, so this basically inhibits, it's an antagonist of GABAergic neurons, which is the interneurons that they were stimulating before, and just random light flicker. So you can see that with no light and with light, here they have this group at 100, significant reduction when you have 40 hertz. Blocked by the addition of picrotoxin, and you don't see any really improvement with, you know, with 20 and 80 hertz. Interestingly, well, I guess that's no improvement. I was going to say a slight decrease with random, but maybe this is a good sign that we're not seeing a significant increase in, you know, and this figure is about the same as this figure. It's just looking at different variants of amyloid beta. I guess it's. Earlier, we saw that, you know, I was seeing that with random flickering, you saw an increase in beta amyloid plaques, but that was optogenetically induced. This one is externally induced. So, you know, maybe randomly flickering fluorescent lights are not so dangerous. I'm extracting maybe too much information out of this. So inhibiting these, so the interesting thing, and this is, I made a note to myself, and I'm going to just blurt this out before I forget it, because I forget these things. Interestingly, adding in a picrotoxin, which is an antagonist of these neurons, which is the orange here, inhibits this response. It'd be interesting to see what happens when you add an agonist of these neurons. Will it boost this response? I don't know. I'm sure there's plenty of drugs out there that are agonists of GABAergic neurons. Okay, so now let's go down to other figures. Now we're looking at microglia, similar to what we saw before, amyloid beta plaque and merging, and we see again all of these bars here basically is just quantified data from these images. This is dark, this is flicker, basically showing the same thing, that the microglia have morphological changes. Their cell body diameter goes up, their processes length goes down, their microglia that have amyloid beta, the percentage of them goes up. So similar to what we saw before. So it appears that with an external flicker of 40 hertz light for so they flickered it for an hour, and then they analyzed, or I guess dissected the mice after an hour of flickering the light. So it's pretty impressive that this was so rapid, that this happens after an hour. So that is to me pretty impressive. And then they looked at again amyloid beta 140, amyloid beta 142. They looked at soluble and insoluble fractions of amyloid beta. I think the insoluble is maybe larger aggregations of them. In either event, it goes down significantly. So we have a significant reduction. And in K, what we're seeing is we're seeing average plaque number. So this is in the dark. This is 40 hertz flickers. This is an amyloid beta hook stain. This is basically nuclei that are staining. Merge. And what's the difference between this one and that one? Well, this is just looking at a whole slice, I guess. This one is looking at just microglia. And the number of the plaque number, actually, average plaque area and average plaque number, and these arrows are pointing to plaques, according to their quantification, goes down significantly. Again, what appears to be about, roughly halved, right? So we're looking at, numbers that are about roughly 50%. So that's pretty significant. That's pretty significant. I'm going to stop sharing right now. The rest of the paper is discussion, and like I said, there's a lot more to it. So just to give you the exact numbers, somebody asked before. Screen share, light flicker decreases plaque load in visual cortex. So hypothesize they would need repeated exposure to affect insoluble amyloid beta. So that one was actually a little longer. So they used mice, and these were these 5x FAD mice, and we're seeing a decrease by 60%, 51%. Okay, so again, about roughly 50% decrease. Now, again, just to kind of reiterate, these are all 5x FAD mice. They do use another animal mouse model, which is this APP mouse model, which is also a model of Alzheimer's. They did not use, correct me if I'm wrong, I don't, they might be somewhere in the extended data, but I don't think they used normal mice that are like very aged, like old mice, right? So I would say there's
probably not. Probably not, to be fair, Oliver, because normal mice don't tend to have Alzheimer's, and that's the problem. And it's one of the criticisms, just to fill people in on who are watching, it's one of the criticisms of using mice as a model for Alzheimer's, because we have to engineer them in specific ways so that they develop plaques just like we do. Some people argue that it's not really Alzheimer's per se, and therefore we can't really make many conclusions based on it. Successful results in a simulated version of Alzheimer's isn't really true Alzheimer's. I'm a bit on the fence really on that. I see what they're saying.
Yeah,
But
I still think. Well, the argument can go. Again, there's way too many variables. Hold on a second. There's way too many variables. You could also make the counter argument that, hey, these are hyper Alzheimer's mice. So if it's working with these mice, it's going to work with the your slower garden variety Alzheimer's. I mean, it's a model, right? I mean, you have to start somewhere, and everything is contrived. You try to control for everything. I don't know if there is a animal out there that's normally gets Alzheimer's that we could use as that normally gets Alzheimer's like big time when it gets old. I don't know.
Well, primates, I believe, do.
Yeah, primates, but something simpler,
Politicians?
Yeah, that's a good one. But that's kind of hard because I think they have early onset Alzheimer's and it lasts throughout their life, so it's kind of hard. Again, that's kind of an artificial model as well.
Yeah, it's not a realistic model of a human being, is what you're saying, Oliver.
Yeah, exactly. I think the mouse model might be a little bit more relevant in this case. Okay. But yeah, these are models, and it's the same with arguments with accelerated aging, mice and also diseases that accelerate aging. My 2 cents is I think these are all valid, and that they're not perfect, but they do represent certain facets of the process. It might be it might be kind of a hyper version, and it not exact. But I think it certainly helps direct us in the right direction, and obviously we're going to do follow-up work. Not us, but maybe us. But there are people out there doing follow-up work with humans because this is a very testable system. I mean, I just went online and got these things, right? And this should be a relatively easy trial to set up, right? I mean, I don't see any difficulty in setting up. If this is a difficult clinical trial to set up, then we might as well just give up, right? On having clinical trials, because I would expect that we see results one way or the other pretty soon, from testing. If we don't see any human results within, I would say, by the end of 2018, that we are doing something severely wrong, and our whole system is defective, which it probably is. So,
But there are a lot of promising approaches, though, aren't there? I mean, in general, not just this light frequency. There's a lot of different.
Yeah.
I'm going to use a cliche, but there's a lot of thinking outside of the box going on, compared to the sort of traditional approaches to things like Alzheimer's. You're seeing things like this light frequency with, all sorts of unusual things where they're modulating the macrophages in different ways, and it's getting interesting results. So I think you're probably right. Within the next few years, we're going to see some very interesting results. Like another example is GAIM game by Proclara, and that was originally it's actually based on, it's a neurophage, and it's based on, it was originally developed at Michael J. Fox for Parkinson's, but they found out that this general amyloid was it general amyloid something motif, I can't quite remember what the I stands for in game, actually clears multiple amyloids and misfolded proteins. So it not only does like tau, beta, and all sorts, but amyloidosis. And I think we're going to see, in the next few years, a breakthrough, and it's going to be on maybe multiple fronts at once.
Yeah. The other thing that is very interesting is that these are 40 hertz light stimulation. But one thing that is also potentially valid is we have other ways to entrain things at 40 hertz that don't use light but use other stimuli that are also just as non-invasive. One of them obviously is auditory. What about 40 hertz auditory tones? And the third is tactile, 40 hertz vibration. So, do these work the same way? Or can they all be additive, having different regions of your brain entrain at 40 hertz? Do they all need to be synchronized at 40 hertz? If they're off by a phase, do they cancel each other? I mean, there's so many, there's so many questions. There's so many great experiments, that follow-up experiments to this. And the fact that all of this can be tested on people, volunteers, non-invasively, I mean, we already live in an environment where we're getting obnoxious, loud noises and vibrations and jackhammers and lights. I mean, this is very, very benign stuff that we're talking about here. And if it could have such a radically positive effect, then it's worth, exploring. And at least finding out whether the answer is yes or no. Does it work or does it not work? I would be willing to bet that it will work. To what extent, I'm not sure. I would be quite surprised that it doesn't work at all. If it doesn't work at all, then how do we explain all of this other data, right? Yeah.
But it might not be 40 hertz for humans. It might be, another frequency. But there's probably a sweet spot, yeah.
Yeah, and I do think it might actually be 40 hertz. And actually, I think for humans, I think it's the range of mammals. It's pretty. They mentioned in the paper it's pretty similar. And I don't want to. I may be garbling this, but I remember talking about this very briefly with Li Wei Sai, this paper, when we were at a meeting. At Harvard at an ancient meeting, and she may have suggested that that having this 40 hertz light stimulation is not localized just to the to the visual cortex, but that it somehow spreads. So that would be very, very promising as well, because clearing out plaques just in the visual cortex is pretty impressive, but if the entrainment actually then spreads throughout the brain, then that's just better. But again, there's a lot of follow-up experiments that are very good. Like I mentioned, and you mentioned, that there's other approaches that can be taken that show that we can boost macrophage clearance, reduce inflammation, and all of these approaches are again generally recognized as safe. And will all of these synergize? Can we have a combination therapy where, you're taking curcumin, you're stimulating yourself with 40 hertz light and tactile vibrational stimulation, and taking some of these other supplements that boost certain positive chemokines? Will this all synergize into a positive treatment? This is something that I'd love to see. Stuff carried out, maybe through the traditional channels of clinical research, but, why not volunteer efforts? Again, it all depends on, if you construct the experiment correctly, then that's really the key. If you have the right controls and you have enough participants and you control for everything, then, of course, with humans, you can't really have a definitive Alzheimer's, diagnosis until you're dead and you take a biopsy of somebody's brain. But, we don't really care about that so much as humans. We just care about the symptoms, right? Really, that's the most important part. And, do you have a reversal of symptoms? And that's something that you can certainly test using memory tests and a number of other types of non-invasive tests. Yeah, so I'm really psyched about this paper for all these reasons.
Yeah, and another possible co-treatment potentially is senolytics as well. You may have read the paper by Goodcoff and his team. They essentially looked at macrophages and they worked out that inflammation causes them to be dysfunctional. Mm-hmm. And when they remove that inflammation, they start working again normally. So they think that senolytics, by reducing the SASP, or the cocktail of inflammatory signals, will improve macrophage, neutrophil function, and the immune system in general. And it probably bears true, because if you recall a few weeks ago when we talked about the gut microbiome, and we talked about its influence on macrophages through inflammation, it all sort of starts to tie up, really. So manage the inflammation, guide the macrophages in how they perform, and the polarity of macrophages as well, because there can be M1 and M2. So I think, I think we're going to make some progress.
Yeah,
we're getting close.
I'm very excited. And, again, I think the difference between, the treatment of diseases of aging and at its root, the whole entropic underlying, basement underlaying of aging itself, that it's going to take a multi-pronged approach, many multi-pronged approach. So it's not this, it's a completely different way of thinking that maybe, I don't want to say post-medical establishment, but there's not going to be an anti-aging pill. I don't think there's such a thing. I think there's going to be an anti-aging, spectrum of stuff that you have, just like to live healthily, you can't just run on a treadmill and say, I've done it, right? You have to have good diet. You have to do multiple exercises. You have to have good social network. You have to have a good, healthy mental outlook on life. And all of this ties into an overall healthy individual. And with something like aging, where you have many things going wrong, you'll probably have to start incorporating, a multi-pronged, therapeutic lifestyle approach, if you will. And right now, the problem is your traditional clinical research trials are not geared to test that, right? They're geared to test one thing at a time. And if one thing takes 20 years, well, I'm not going to wait 2,000 years before, I get approval of a multi-spectrum approach. So there has to be, I think, a whole reevaluation for how these trials are structured.
Yeah, I mean, combinational testing is quite challenging. I mean, I know that through the work we've done with mice about, we've looked at the, it's mind-boggling the amount of potential issues and noise. And you have to have a lot of mice to get rid of the statistical noise if you're going to do that. We have a question. Dwayne here has asked, do they distinguish between resident microglia and migration of monocytes, macrophages from the periphery?
Good question. I don't know is the simple answer. I didn't read through the extended data, but I can go back to the paper and see what they used as a marker, and that marker might be indicative of which of the two categories they are or not. I'm just going to quickly scan through this paper and see what they used. I
mean, in sort of related or linked experiments, like by Stolzing, etc., they do see that it's actually the microglia that's, the tissue-resident macrophages that are actually, being reactivated. But,
Just going through surgical procedures, radiology,
but they're finding out more and more about how the immune system is so intrinsically linked to aging and tissue upkeep. It's like there's a new discovery almost every week. I swear, every week there's just new stuff being found out. Like a couple of weeks ago, they found out that neutrophils have a major role in repairing neurons and things like that. And they're very critical in clearing away cell debris to facilitate repopulation with fresh cells. So they thought it was macrophages, but it was neutrophils. But the interesting thing is, they removed neutrophils and they found macrophages picking up some of the slack. So there's quite a lot of redundancy built into the immune system. And it's almost certainly the immune system is a big key to aging, almost absolutely without any doubt. So if we can crack those secrets, we're on a winner there, I think.
Just looking at particular antibody.
I see my Mike Lustgarten's just joined us in the chat there. He was probably his spider sense was probably tingling when I talked about microbiome.
So in the materials and methods, I think what they use to detect the microglia is a antibody against IBA1, which is a microglia / macrophage specific protein. So maybe it is not differentiating between either of the two. So very good question. So there you go. If they're using an anti-IBA1 antibody.
So it could be either or both.
Yeah.
So I mean, it could be encouraging macrophages from outside to migrate, to the area of interest.
Well,
that certainly happens in other tests that they've run.
Well, if I'm reading it correctly, that seems to be the case.
It was certainly the case with experiments they did with arterial plaques and other things like that. So, yeah. What we need to do now is create a superhero version of our normal macrophages, and we're sorted, because that's another avenue that people are actually exploring. Some researchers are trying to make macrophages much, much more durable and resistant to things like inflammatory signaling, so that they don't become as dysfunctional, so that they can actually keep doing the job they're meant to be doing for longer, which kind of makes sense. Again, another potential co-therapy.
Oh, here's my idea for like a super therapy. You create a super cell, right? That's a macrophage that can do all of this, migrate everywhere, eat senescent cells, and then once it's at the right location, turn into the proper somatic stem cell. Now you gotta design this very, very right, because you can actually create a super cancer at the same time. Anyway, just tossing it out there. Super cell.
Well, yeah, you say that, but they it's interesting because they're already starting to design custom cells. They made I covered it a couple of weeks back on the actual blog. They were actually engineering a type of T cell, another kind of immune cell, but it wasn't. It was from normal cells. They just took, I think it was liver cells, and they actually changed them into a different type. They created custom T cells, and what was interesting was with these T cells, they put longer probes on them, so that they were able to detect cancer from further away, and they created almost like a little gene circuit. I cover it on the blog, and they created like an either and or. It had to have two conditions met in this circuit, like an AND gate. I think it was an AND gate or an OR gate. I think it was an AND gate, and it worked exactly like that. So this custom T cell spots cancer a lot more efficiently than normal T cells, and it's not even an immune cell originally. So your idea is not that out there,
So yeah, so it could be either a cocktail of supercells or one supercell, and it basically what we're talking about here is reverse engineering a nanomachine, right? Because if people talk about injecting nanobots into the body, well, that's what a cell is. You're not going to have this thing made out of titanium floating around. I've seen these artists' renderings of some sort of like, R2-D2 robots floating through your bloodstream. I mean, nanomachines exist. They're cells. They're ribosomes. They're based on macromolecules that are biological. So in this sense, what we're seeing right now, perhaps with this whole cell therapy, which is a very, very early form, super primitive early form, but very promising of our development of nanomachines that are basically super cells that will go into the body and rebuild ourselves from.
Well, I've said this before. If we ever manage to get to the point in nanotechnology soon, where we can literally engineer nano immune cells that are multifunction, we might as well just turn the lights off and go home, because it'll solve so many problems. You'd be able to get rid of crosslinks with nano machines, and clear amyloids and spot treatments, and deal with cancers. It's very much on the list of things we would very much like to have in biology, but it isn't there yet. But honestly, I think if they do crack it, then I think it takes a huge chunk out of the difficulty of bringing aging under medical control. Definitely.
Well, lots of other
wish list.
Lots of other things. Tissue damage, just slather on some super cells.
Super cells. But, well, yeah, they are doing stem cell bandages here in the UK.
Yeah.
They're actually testing them. Yeah, it's very interesting.
Sure.
And, of course, Japan has introduced new laws to encourage the development of stem cells as drugs. They're very keen on that. So I think, it's just very exciting. All of it, really. I'm not so much into sitting under energy triangles to try and top up my chakra, but I'm all for unusual or unconventional approaches, as long as they're obviously grounded in science. And this is very interesting.
Right. So this is the only energy therapy, potential therapy that I'm aware of that is pretty well grounded. So. I don't know. I have nothing else to say except stare at the orb of longevity, everyone.
I can feel my mental cognition improving just from looking at it for that short period of time.
Really?
Yeah. Although we've got lights like that on our Christmas tree we've just put up downstairs, and they sort of glow like glowworm lights.
Mm-hmm.
Maybe I'm going to go and have a look at that for another hour or two and see how I feel. Yeah. But it does beg the question, though, doesn't it, Oliver? Because looking at the random flickering pattern, that's a bit concerning, really. But I suppose it makes sense, because if it's randomly firing these genes,
it's probably suppressing 40 hertz. It's probably suppressing it. Yeah.
It's then going to sort of deregulate everything. So I suppose it makes sense that the situation becomes worse if it's randomized. But then it makes you think, TV monitors or computer monitors, what do they really do?
Well, here's a thought. Has anybody done an epidemiological study to see if more people are going insane working under fluorescent lights that are flickering like crazy?
I'm sure I've seen some data for fluorescent lights. I'll have to have a look, but I'm sure it's not very conducive to your health, in all fairness.
Perhaps we're not. Hopefully, we're not giving any ideas to people constructing secret CIA torture prisons to. Oh, let's just deregulate those fluorescent bulbs, drive our prisoners more crazy.
It's possible.
It's possible. But with every technology, it's dual purpose. So we want to do good, not bad, people.
Yeah, any technology is potentially subject to abuse, isn't it? It's like a lot of people. One of the concerns that people sometimes have about rejuvenation biotech is its potential to be abused. But
everything is.
Exactly. I mean, even electricity and all sorts could be abused. But just think, if it's in the right hands, the rewards are potentially vast.
Yeah.
I mean, who wouldn't want to see the end of things like cancer, Alzheimer's, Parkinson's? I think it's worth a risk.
Of course, that's what life is all about, isn't it? Risk.
Yeah, risk versus reward, and the rewards are potentially vast. So
they are indeed.
Okay. So there you go.
Well, Merry Christmas, everyone! Happy Kwanzaa, Happy Holidays, and happy, I don't know,
Hanukkah,
Hanukkah,
Diwali,
and
or whatever's going on.
and yeah, I apologize to any sect that I have omitted.
So, yep, that was great. Anyway, very interesting. Very interesting paper, and hopefully everybody enjoyed that. Sorry about the technical fault about halfway through there. I think we recovered okay, didn't we, Oliver, in the end? We sort of held our own there, so that's great. Well, thanks for tuning in, and we'll be back in the new year, so enjoy your holidays, stay safe, and thanks very much to the Lifespan heroes who make this monthly journal club possible. And if you want to learn a bit more about us, check out our website, www.lifespan.io, and wish you a happy new year as well while I'm here. And thank you very much for tuning in.
Yep. See you in 2018.
See you next year. Bye.